Cancers occur when cell replication goes out of control, forming lumps of cells called tumours. If we can stop these cells from replicating, then we can help to treat some forms of cancer. Cisplatin, which is the molecule shown in the centre, is an anti-cancer drug which can be used in chemotherapy. Cisplatin works by binding to and distorting the structure of DNA. Now, DNA usually exists as a double helix, but when cisplatin binds, it loses its two chlorine groups. It then forms bonds to the guanine bases on the DNA. This binding forces the DNA to kink into this irregular shape. This stops the DNA from unwinding and replicating properly. By stopping DNA and cells from replicating, the cancerous cells die. So here we can see the cisplatin bound to the guanine bases in DNA, and it's this kinked structure which is typical of cisplatin. Now cisplatin has two chlorine groups. They're shown in green next to each other. There's an isomer of cisplatin called transplatin, which has the two chlorines opposite each other. Now if the isomer transplatin was used instead, then it couldn't bind properly as the chlorines are in the wrong position to be replaced with bonds to the DNA. This is why it's important to be able to differentiate between the two isomers. Even though they have the same molecular formula, the specific organization in space is key.